Welcome to the course on medical nanotechnology. In this module, we will explore the fascinating field of nanoparticle-based antiviral drug delivery systems. Nanotechnology has revolutionized the medical field by offering unique opportunities for targeted therapy, enhancing drug delivery efficiency, and overcoming various challenges in antiviral treatments. In this script, we will delve into various aspects of nanotechnology and antiviral therapy, focusing on topics such as optimization of physicochemical properties of antiviral nanoparticles, surface functionalization strategies, multifunctional nanoparticle design, and many more. Antiviral drug delivery systems based on nanoparticles have shown promising potential in enhancing drug efficacy and reducing side effects. In this topic, we will discuss the crucial role of optimizing the physicochemical properties of nanoparticles to achieve efficient drug delivery. We will explore parameters such as particle size, surface charge, shape, and composition, and their impact on drug encapsulation, release kinetics, and stability. Additionally, we will delve into the methods employed to precisely control these properties, such as bottom-up and top-down approaches, self-assembly techniques, and the use of various materials like lipids, polymers, and metals. One of the key challenges in antiviral therapy is achieving targeted drug delivery to specific viral sites while minimizing off-target effects. Surface functionalization of nanoparticles offers a powerful approach to achieve this goal. In this section, we will explore various strategies to modify the nanoparticle surface by incorporating ligands, antibodies, peptides, or aptamers that can specifically recognize viral antigens or cellular receptors. The use of targeting moieties can enhance the accumulation of nanoparticles at the desired sites, leading to improved antiviral efficacy. We will also discuss the importance of selecting appropriate targeting ligands and the potential for active and passive targeting. Combination therapy, involving the use of multiple drugs with distinct antiviral mechanisms, has gained significant attention as a promising approach to combat viral infections. Nanoparticles provide an excellent platform for designing multifunctional systems capable of co-delivering multiple drugs with precise release profiles. In this topic, we will explore different strategies to engineer multifunctional nanoparticles, such as layer-by-layer assembly, core shell structures, and dendritic systems. We will also discuss the benefits of combination therapy, including the reduction of drug resistance and synergistic effects, as well as the challenges associated with optimizing drug ratios and release kinetics. Vaccines play a critical role in preventing viral infections and inducing long-lasting immunity. Nanoparticle-based vaccine delivery systems offer several advantages, including enhanced antigen stability, controlled release, and targeted delivery to antigen-presenting cells. In this section, we will explore different types of nanoparticle-based vaccine platforms, such as liposomes, polymer nanoparticles, and virus-like particles. We will discuss their potential for delivering viral antigens, adjuvants, and immune stimulatory molecules to elicit robust immune responses. Gene silencing techniques, such as RNA interference, RNAi, have shown tremendous potential in suppressing viral replication and gene expression. Nanoparticles can effectively deliver RNAi molecules, such as small interfering RNA, siRNA, or short hairpin RNA, shRNA, to target cells and tissues. In this topic, we will explore the design principles and mechanisms of nanoparticle-mediated gene silencing approaches. We will discuss various nanoparticle formulations, such as lipid-based nanoparticles, polymeric nanoparticles, and inorganic nanoparticles, and their ability to specifically deliver RNAi molecules to inhibit viral gene expression. The blood-brain barrier, BBB, poses a significant challenge in the treatment of viral infections affecting the central nervous system, CNS. Nanoparticle-based drug delivery systems offer unique opportunities to overcome this hurdle and deliver therapeutic agents across the BBB. In this section, we will explore various strategies to engineer nanoparticles capable of crossing the BBB, including coding modifications, receptor-mediated transcytosis, and focused ultrasound-mediated disruption of the BBB. We will also discuss the importance of selecting appropriate drug cargoes and the potential risks associated with nanoparticle-mediated drug delivery to the CNS. Emerging viral infections, such as the Zika virus, Ebola virus, and Middle East Respiratory Syndrome, MERS, coronavirus, pose significant threats to global health. Nanotechnology offers innovative solutions to combat these emerging infectious diseases. In this topic, we will discuss the application of nanotechnology in developing antiviral strategies against emerging viruses. We will explore the use of various nanomaterials, such as gold nanoparticles, quantum dots, and carbon nanotubes, in diagnostics, therapeutics, and vaccine development. Additionally, we will examine the challenges and future prospects of nanotechnology in addressing emerging viral threats. In nanoparticle-based antiviral therapies, the interactions between nanoparticles and the host immune system play a pivotal role in determining therapeutic efficacy and safety. In this section, we will explore the complex interplay between nanoparticles and various components of the immune system, including immune cells, cytokines, and complement proteins. We will discuss how nanoparticles can modulate immune responses, either by enhancing antiviral immunity or suppressing excessive inflammation. Furthermore, we will examine the factors influencing nanoparticle immunogenicity and strategies to minimize adverse immune reactions. As we harness the potential of nanotechnology in antiviral therapies, it is crucial to understand and mitigate potential toxicological risks. In this topic, we will explore the field of nanotoxicology, which focuses on understanding the interactions of nanoparticles with biological systems and evaluating their safety profiles. We will discuss the various factors influencing nanoparticle toxicity, such as physicochemical properties, surface modifications, and biodistribution. Moreover, we will delve into the immunotoxicity considerations associated with nanoparticle-based antiviral therapies, including hypersensitivity reactions, immune tolerance, and long-term effects on the immune system. Viral escape, characterized by the emergence of drug-resistant strains, poses a significant challenge in antiviral therapy. Nanoparticle-based drug delivery systems can be engineered to address this issue and minimize the development of drug resistance. In this section, we will explore different strategies to prevent viral escape from nanoparticle-mediated therapy. We will discuss the use of combination therapies, RNA-based approaches, and immune-activating nanoparticles to target different stages of the viral life cycle and reduce the likelihood of resistance development. Real-time monitoring of therapeutic response is essential for optimizing treatment strategies and assessing treatment efficacy. Nanosensors, integrated within nanoparticle systems, offer the opportunity for non-invasive monitoring of antiviral therapy. In this topic, we will explore the development of nanosensors based on various principles such as fluorescence, surface plasmon resonance, and magnetic resonance. We will discuss their applications in monitoring nanoparticle biodistribution, drug release kinetics, therapeutic response, and potential challenges in translating these sensors into clinical practice. Despite the tremendous potential of nanoparticle-based antiviral therapies, several challenges need to be overcome to translate them into clinical practice. In this section, we will discuss the translational challenges associated with the development, scale-up, and clinical translation of nanoparticle-based antiviral therapies. We will explore the regulatory aspects, manufacturing challenges, and clinical trial considerations. Moreover, we will address the importance of interdisciplinary collaborations, intellectual property protection, and the economic viability of nanoparticle-based therapies. In conclusion, the field of medical nanotechnology offers unprecedented opportunities for revolutionizing antiviral therapies. Nanoparticle-based drug delivery systems have the potential to enhance drug efficacy, improve targeting, and overcome numerous challenges in treating viral infections. Through the optimization of physicochemical properties, surface functionalization strategies, multifunctional design, and the interplay with the immune system, nanoparticles can be tailored to meet the specific requirements of antiviral therapy. However, significant translational hurdles and potential toxicological risks need to be addressed to bring these promising technologies from the laboratory to clinical applications. With continued research and collaborations, medical nanotechnology holds tremendous promise in combating viral infections and improving patient outcomes.